similarly, PD1, when bind with the PD1 ligand, it gives the inhibitory signals. And this field has been used in a very good way for the cancer treatment, and people have used those monoclonal antibodies or agonistic antagonistic antibodies against those or blocking antibodies against those immune checkpoint molecules to treat the patient. So what we found that when we took the VL patient's blood, and when we went for the transcriptional analysis of CD4 T cells, we found that the LAC3 is upregulated. So LAC3 is basically it combines with the MSC class 2 molecules, and it gives negative signals in both APC and in the CD4 T cells. And you can see here that VLCD4 T cells have more expression of those LAC3 molecule compared to the post-treatment and endemic control. And most importantly, again, we wanted to see whether there is any therapeutic potential of this knowledge. We know that there is expression of those inhibitory molecules on the CD4 T cells, but can we manipulate it for the host-directed therapy? So what we found that when we block this type 1, when we block this LAC3 molecules using blocking antibody, we found enhanced antigen-specific interferon gamma production. Someone asked a question this morning that why the vaccine against malaria is not very successful. It's just one of my viewpoint is that what happens that, for example, RTSS vaccine, which has been tested in healthy volunteers, NAV volunteers, it worked very fantastically. It shows efficacy of around 70 to 100%. But when it was tested in the field setting, primarily with the children living in the Africa, that efficacy went down and it came around 30 to 50%. One of the possible explanation is that the people who are living in the field, they are continuously exposed to that antigen or that parasites. And in response to that, they already have imprinted an immune regulatory network. So that when they are vaccinated, the vaccine-mediated anti-parasitic immune response is suppressed. So one need to be very careful while designing those vaccines and should consider how this vaccine can manipulate the established immune regulatory network during the infection. So just to conclude this part, what I tried to say is that Th1 cells, once activated during the infection, they produce interferon gamma, TNF alpha. This gives signals to macrophages to kill those parasites. However, if this signal is blocked, parasite they get an opportunity to multiply, divide, and persist in the host. And one of the important player is the TR1 cell, which produces IL-10, and this IL-10 suppresses the activity of both phagocytic cells, antigen-presenting cells, as well as the CD4 T cells. And one of the important player in driving this TR1 cell IL-10 production is the type 1 interferon, which comes from the dendritic cells in the macrophages. But not only this, apart from that, these inhibitory receptors, which are expressed on the CD4 T cells as a part of homeostatic mechanism due to continuous exposure to antigen, are also very critical. And then the transcriptional repressor like BLIMP1, aryl hydrocarbon receptor, etc., which drives the IL-10 production by Th1 cells. So thank you everyone for your kind attention. I can't hear you. Mute yourself.
Sorry, can you hear me now?
Yeah, I can hear you now.
Yes. So there are two questions. One question, the first question is, how can a vaccine be devised against a pathogen which directly inhibits the activity of macrophages? Or how a prophylactic formulation can be devised so as to target APCs?
Well, so to answer this question, as I told, like I gave the example of RTSS vaccine. So we know that this antigen presenting cells, they do express the immune checkpoint molecules on their surface, and they do show their inhibitory effect. So I'm not sure, but maybe when we are going to formulate a vaccine, can we use those antibodies or the blocking antibodies as an adjuvant, which can specifically blocks the activity of those molecules, but then it drives a clear expansion of effective Th1 response. So that can be one way to design a vaccine for the proper activity.
H1 cells?
Actually, we are doing another experiment in the mouse model, and we are trying to see whether type 1 interferon can activate the expression of inhibitory molecules or not. So far, we don't have any idea that whether type 1 interferon is driving the expression of those molecules or not, but we know clearly that those type 1 interferon drives the expansion of IL-10 production by TR1 cells. But yeah, we are doing some experiment to know whether those IL-10 producing TR1 cells are also have high level of LAC3 expression or TIM3 expression or even TGT expression. That we have some data, but I cannot say anything right now.
The next question is that does interferon gamma, which shares the receptor with the interferon alpha, that is the interferon R1, R2 receptor, also suppresses anti-leishmanial immunity?
No. In fact, we have shown clearly that we took the splenic aspirate cells, and when we blocked type 1, when we blocked interferon gamma, we saw very increased number of parasites in those splenic aspirate culture. So clearly, interferon gamma is very critical. So what is happening, even those people who have established leishmania infection, they constitutively produce interferon gamma, and that gamma is critical for controlling the infection. The only reason is that the immunosuppressive environment is not allowing those interferon gamma to show its proper function. So we need to be very careful. Interferon gamma is very critical for controlling the infection, and not only we, but many other groups have shown in animal model as well as in human visceral leishmaniasis that in absence of gamma, parasite number goes up.
So would you say that there will be a benefit if you try to block the TR1 cells or the IL-10 from the TR1 cells?
Yes, so I saw one of the data where when we blocked the IL-10, we saw the reduction in the parasite number, and at the same time we saw enhanced TNF alpha and interferon gamma production by Th1 cells. So yes, I mean, in fact, in 2010 we submitted. So we submitted a grant to NIH based on that study to use IL-10 inhibition in the VL patient, but due to the regulatory, authorities in India, and then we could not conduct that trial. So, but again, you need to be very careful, because when you are going to block IL-10, what happens in this disease model? IL-10, it suppresses function, but again, it is required to control the TNF-mediated tissue pathology. What we have seen that in absence of IL-10, TNF, it destructs the pathology, it destructs the architecture of spleen cells in a very huge way. So IL-10, although it suppresses, but it also protects from tissue-mediated pathology caused by TNF. So you need to be very careful what amount of IL-10 you require to prevent the tissue damage, and what amount of IL-10 is required to control the parasite. There should be a balance. I mean, you can.
Sorry. So here in this case, the TR1 cells are basically the induced Treg cells.
Yes, they are FOXP3 negative.
Yes. So my what I would like you to comment is, like, if you are able to specifically block the TR1 cells and not the natural Treg cells, which also produce IL-10, so maybe this kind of a balance can be arrived. What would you?
Exactly. That is what I told, that when you are going to use those vaccine in the field where you have established immune regulatory network, you need to consider these points also. And yeah.
There's one more quick question from the audience, and after that, I need to ask one question related to the vaccine. So, one of the participant wishes to know whether any similar work has been done in the neurocysticercosis cases, and would ruxolitinib work in such cases?
I don't know about that, but ruxolitinib has been used in myelofibrosis. It has also been used in type 1 interferon adenopathy. So, I mean, it is a well-known orally administered drug. So, yeah.
So, to contribute information towards a vaccine design in these parasitic infections. So when you use these blockers like the JAK1, JAK2 blocking agent along with the other known inhibitor. So did you see whether there is any enhancement in the development of memory T cells? Because that is what is required when you want a vaccine to work.
Yes. So yes, we have seen in the experimental model of VL, once we block, we do see enhancement of the memory T cell response. Yes.
Okay. Thank you very much, Dr. Rajiv Kumar. And if there are no other questions from the panelists, any questions?
I need to go. I have a meeting scheduled from 3 o'clock, so maybe I make a pardon.
Thank you very much. Thank you for a wonderful talk. And you may log out now, Dr. Rajiv Kumar, and we go on to the next talk by Dr. Preeti Desai. She is currently the head of the Department of Biological Science and Technology, Institute of Advanced Research, Gandhinagar. And she's basically working as a scientist in Cell and Molecular Biology Department of B.V. Patel. She was earlier working as a scientist in B.V. Patel PERD, that is the Pharmaceutical Education and Research Development Center, Ahmedabad, Gujarat. And she's a recipient. She has received a PhD degree from Bhavnagar University and was also associated with Cadila Pharmaceutical Limited before joining PERD Center. And she has completed her postdoctoral research from National Institute of Pharmaceutical Research, Ahmedabad, and was a recipient of the DBT Research Association. She has more than 19 years of experience in research and 10 years of teaching experience. And today, her research interest mainly lies in bacterial pathogenesis and development of prophylactic against enteric pathogens, mainly for Shigella and Salmonella. And she has published several articles in these area of vaccine design and development in various recognized international journals. And she is a recipient of several national and international travel grants, and has also received various government funding for her research. So quickly to go on, the title of her talk is Vaccine Development Against Enteric Pathogens. What are the current challenges and future prospects? So Dr. Preeti Desai, can you please go ahead with your talk? Thank you.
Thank you, Dr. Kalpana. And I once again like to thank Dr. Sunil Arora and Dr. Amit for giving me the opportunity to present my talk. Okay. So my today I am going to discuss about the development of vaccines against the enteric bacterial pathogen. Here I main emphasis only on the bacterial pathogen and particularly by the mucosal way, or that is the oral mucosal vaccines. So my talk is a little bit different from the basic immunologist. I am not going to discuss much about the basic immunology, and I am sure that by Dr. Amit Awasthi talks, Rajiv's talks, and the yesterday's talks, the audience have a basic background of the B cells and T cells and the differentiation of the B and T cells. So quickly, when any pathogen enter into our system, particularly in the mucosa, our body has a different layer of the mucosa. And because of this physical barrier, the pathogen cannot make easily entry into our system. For example, if by chance they enter into the gut system or any epithelial lining, the epithelial cells are tightly packed. So because of this tight junction, they cannot enter. Apart from that, this epithelial cells has a specific structure. For example, they have a villi. So again, they cannot enter. The another one is in this mucosal lining, there are certain cells, for example, globally mucosal producing cells. So this continuously secrete the mucus layer. This mucus layer are quite viscous. So if any pathogen enter, they enter up into the mucus layer and destroy. Apart from that, there are certain cells in the enterocytes which continuously secrete the antimicrobial. So this is the first line of the defense. If pathogen enter, and because of the presence of the certain pathogen-associated molecular pattern, our immune cells come into the picture. For example, macrophage. So macrophage first taken up this kind of the pathogen and try to phagocytosis and destroy it. But during this process, the inflammation occurs. And because of that, the adaptive immunity come into the pictures, and adaptive immunity is a specific immunity which generates specific memory B cells and T cells. And for the vaccine purpose, we are main emphasis on the adaptive immunity. So briefly, we all know about the humoral immunity and the cell-mediated immunity, but in general, the antibody are required, or the function of the antibody is to prevent or reduce the infections by clearing the extracellular pathogen through binding to the enzymatic active site of the toxins or preventing their diffusions. Many enteric pathogens enter into our system and they secrete the toxins. So antibody bind to this toxin molecule and diffuse them. The second one is they prevent the binding and entry of the pathogen to the host cells, as well as they also promote the opsophagocytosis of the extracellular bacteria and enhance their clearance, and also activate the complement system. We have a different class of the immunoglobulin in our system, and predominantly immunoglobulin found in a different part of our body is based on their functions. For example, I am more emphasize on the IgA. IgA is a secretory antibody which is mainly found in the mucosa-associated lymphoid tissue or in the mucosal lining. On the other hand, the serum, the IgG is predominantly found in the systemic immunity. Cellular immunity, again, two type of the T cells, CD8 and CD4 T cells. I am not going into the detail about this cells because I think most of the speaker has described about the function of the different cells. But generally, Th1 is mainly helpful for the intracellular pathogen, and Th2 is mainly used for the extracellular pathogens. And my main focus is the mucosal immune system. So mucosal immune system is also called as a local immune system, or the local sites where we require to produce the immune system, in contrast to systemic immune system. So mucosal lining has a specific structure. They are covered with the single layer epithelium, which are covered by the mucus and antimicrobial peptides. This mucosal immune system also has a specific function. For example, they are separated in two sites. One is an inductive and another one is an effector site. So in inductive sites, the immune response induce, while in effector sites, they generate or they protect. And these two sites are anatomically and functionally different. And immune cells are generally migrate from the inductive sites to the effector sites by using the lymphatic system or circulation system. Okay, so as we are main focus, my talk is main central to the enteric pathogen. I am more talking to the gut mucosal immune system. Other than the gut, we also have a nasal mucosal system, or the nasal associated mucosal tissue, which we call as a NAT, and BALT, that is a bronchial associated lymphoid tissue. But enteric pathogen mainly enter through the fecal oral route and establish the infection at the intestine. So we are talking about the gut mucosal immune system. Main function of the all mucosal associated lymphoid tissue are remain same. The gut is the largest immunoglobulin organ in the human body. The intestinal mucosa of an adult roughly contain more than 80% of the body's activated B cells, and that main portion is the IgA productions. This gut lining or the gut mucosal immune system protect the host against the microbial pathogens, and they play a very critical role. They balance, once they provide immunity against the pathogen and also induce a tolerance for their microbial flora as well as the food allergy. So it play a very important role between a balancing between the two type of the antigens. So briefly, what is the architecture or what are the cells specifically present on the gut or the gut associated lymphoid tissue? So generally this is a gut or the gut associated lymphoid tissue. It has the AM cells. So AM cells is a specific kind of the cells which is we call as a microfold cells, and presence of certain AM cells in a particular region is known as a Peyer's patches. So AM cells has a specific property. It does not have a microvilli like other enterocytes have, as well as no mucus layer. And because of this, most of the pathogen take advantage to enter through the AM cells. Now, exactly at the below of the AM cells, at the basal side of the AM cells, we have a follicle associated, we have a dome in which we have a T and B cells follicles. Now, again, exactly to adjustance to this Peyer's patches or the gut, we have a lamina propria. So generally immune response induced at this place, then migrate to the through the circulations, and effector site is a lamina propria. So what happens exactly when any antigen or pathogen enter into the gut system or gut epithelium? So these antigens are firstly taken by the AM cells which are present on the follicle associated epithelium. Now after transcytosis to this antigen, the dendritic cells or the specific professional antigen presenting cells taken up these antigens and hand over these antigens to the T cells follicle or T cells. Now, the antigen which are primed with the T cells which are primed with this antigen are produced or differentiated into the IgA positive B cells with the help of the certain class switching as well as the presence of the cytokine like TGF beta, IL-2, IL-5, IL-6 and IL-10. Now this IgA B cells or the B cells which are rich on the IgA part are also have a mucosal imprinting. So this mucosal imprinting molecule like CCR9, CCR10 and alpha 4 beta 7 integrin make them to seed back to the lamina propria. Now this molecule go to the blood circulations, and from the blood circulations, they again differentiated into the plasma cells with the help of the antigen-specific Th2 cells, which produce IL-5, IL-6, and IL-10. Now, this again plasma cells, which produce the dimeric IgA, are attached to the basal site of the epithelial cells through the PIGR receptor, that is a polymeric immunoglobulin receptor. And after attachment to these sites, they secreted out from the lamina propria as a secretory IgA antibody. So why we are so much emphasize on the secretory IgA antibody and what is the role into the enteric pathogen and mucosal immunity? So secretory IgA antibody is a predominant antibody found in the most of the secretions, including in the gut. It is more than 70% of the all total IgG antibody. IgA is also resistant to the protease enzyme, which in the case IgG is not. It prevents attachment and invasion of the enteric pathogen, particularly at the site of the infection, and also neutralize the enterotoxins secreted by the enteric pathogen. There are two types of the secretory IgA, IgA1 and IgA2. The secretory IgA is also produced by the two pathway. One is T-cell independent pathway and another one is a T-cell dependent pathway. So T-cell dependent pathway which produces secretory IgA remain longer period of time, and it is mainly induced by a vaccine. In general, the secretory IgA can prevent or induce two types of the immune response. It's like a low affinity, innate like low affinity response, as well as infection and vaccines induce high affinity protections through the invasion. So they prevent the pathogens to invade into the epithelium. But if pathogen enter into the epithelia, then the polymeric IgA and IgM exert the non-inflammatory effects inside the epithelial cells and neutralize their toxins. But again, if any pathogen or antigen cross this epithelia and enter into the cells, then secretory IgA or IgA cannot do anything. And to prevent such kind of the infections or inflammation, we require serum IgG. So why we are so much emphasized on the mucosal immunity? If you see, most of the vaccines which are available in the market or licensed are mainly injectable based. There are very few vaccines which are available for the oral use or oral mucosal vaccines, namely hardly 2 or 3. For example, polio vaccines or cholera or the typhoid vaccine. So the problem with this design of the mucosal vaccine is it requires a specific formulation. It should be survive in our intestine system, as well as it also produces effective immune response. And most of the enteric pathogen are entered through fecal oral route and establish their infection at the intestinal sites. So the mucosal immunity which are generated at the intestinal sites are very beneficiary to protect against the enteric pathogens. Now, so what are the enteric infections? So basically enteric infections are caused by the consumption of the contaminated food and water, and it's mainly characterized by food poisoning and infectious diarrhea. Roughly, diarrhea kills approximately more than 2,000 to 3,000 children every day, and which is more than combined AIDS, malaria, and measles. It is second leading cause of the death among the children under the age of 5, and this disease not only kill the children or it's highlight for mortality, but it also causes the morbidity, because frequent diarrhea can hamper the growth of the children also, or their brain development and learning ability. Among the enteric infections, protozoa, virus, and bacteria are the main causative agent of the enteric infections. And bacteria are responsible for the 48% of the cause of the enteric infections. Now, among the bacteria, the cholera, Shigella, non-typhoid salmonella, and Enterobacteria enterosia are the major bacterial pathogens which cause the diarrheal disease. And unfortunately, we have vaccines against cholera, but it's only effective to the low efficacy. We also have vaccines against Salmonella typhi, but we don't have vaccines against Shigella, ETEC, and non-typhoid salmonella, which are a major cause of the diarrheal disease. Although the cholera vaccine, which has a cholera toxin B subunit, gives some kind of the protection or cross protection to ETEC, but we still not have a single vaccines against ETEC. Now, the question then comes, because this is a bacterial pathogen, so we can simply prevent this bacteria by just preventing or by giving the basic need of the human being, for example, clean water, proper sanitization, and the hygiene condition. But as we all know that it's a very challenging condition, particularly in the developing and underdeveloped country, where the most of the people are live in the crowded place. They do not have clean water for their drinking purpose, and other hygiene conditions are also questionable. So it's not possible to give by simply implying this basic sanitizations. Normally, antibiotics are also given for the treatment of the bacterial infections, but day by day, antimicrobial resistance become a threat, and it's estimated that by 2050, most of the bacteria acquire the resistance to the available antibiotic. It is also estimated that around 90% of the Shigella strains are currently resistant to the previously used antibiotic. And WHO also classified all pathogen based on the antimicrobial resistant threat to give the higher priority for the vaccine development. And they classified like critical stage, high and medium, and E. coli, Salmonella and Shigella, all three are there in the WHO list. So to fight against the antimicrobial resistance, vaccine is one of the best options. But with the help of the various government agency and the various non-government agency, we don't have any vaccines against Shigella, etc., non-typhoid, Salmonella. Although there are a number of efforts have been made to develop the vaccine, but they are just reached to either stage 2 or stage 3 trial, but there are no vaccine available in the market to treat these infections. Now, to develop any vaccine, it is very essential to understand the pathogenicity of that pathogen. So we just briefly discuss about the pathogenicity of the enteric bacteria, and we mainly focus on the 4 bacteria: Vibrio cholera, Salmonella, enterotoxic E. coli, and the Shigella. I have divided this 4 organisms into the 2 categories. One which produces a disease by making the invasion, that is an invasive pathogen, and one which causes the diarrheal disease just by secreting the enterotoxins. It's not invade into the mucosa, and that is the non-invasive. So non-invasive pathogen are the Vibrio cholera and ETEC, that is the enterotoxigenic E. coli, while invasive are Salmonella and Shigella. So non-invasive pathogen colonize the intestinal mucosa without the invasions and secrete their toxins, which leads to watery diarrhea. While invasive pathogen mainly use M cells for their portal of entry. For example, Salmonella translocations of the intestinal mucosa and then spread into the bloodstream and systemic other organ. Like Shigella do the local invasion, inflammation, and they spread from one epithelium to another epithelium and cause the destruction of intestinal mucosa. E. coli, for example, enterotoxic E. coli, ETEC, with the help of special colonization factor, it attached to the epithelial cells. And after attachment to the epithelial cells, it has a 2 subunits, 2 toxin, heat labile, that is LT, and heat stable toxin. The LT toxin is mainly responsible, or major portion of the LT toxin is mainly responsible for the diarrhea. Now, LT toxin has a 2 subunit, A and B. So B subunits binds to the GM1 receptor on the membrane, and A subunits then liberated from the B subunits and causes the or bind to the protein which are involved in the regulation of the cyclic AMP. And because of this binding, the cyclic AMP regulation is hampered, or cyclic AMP increasing, which leads to secretion of the minerals from the cells and cause the diarrhea, watery diarrhea. Vibrio cholera has the same function. Again, same type of toxin, B subunits binds to the membrane receptor and A cause the reactions and do the watery diarrhea. In this case, Vibrio cholera also causes the formation of the biofilm in some times. Now, what about the Shigella? So to explain the Shigella pathogenicity, in our lab, we have created one animation. So I hope this animation will give you a clear picture. So when Shigella enter into our system, it first encountered by the M cells and enter into the M cells. Then this M cells hand over this Shigella to the macrophage, which are present on the basolateral sides. Shigella induces the apoptosis and lyse the macrophages, and once it lyse the macrophages, it enter into the adjacent epithelial cells and through one cells to another cells, making the secrete by making the acting like filament, and this causes the disturbance of the epithelial lining or destruction of the epithelial lining, which causes the mucus and blood in the stool, and it's characterized by the dysentery. So this about the Shigella. Salmonella has a similar mechanism, only difference is here in macrophage, it remains in the vacuole and then they spread systematically. Okay, so any vaccine which produce against this four kind of the bacteria, this vaccine should induce protective immunity by preventing the infectious agent from attaching to and colonizing the mucosal epithelium, particularly in case of the non-invasive bacteria, through secretory IgA, by blocking the binding of microbial toxins to the epithelium and other target cells, again probably via secretory IgA. Vaccine should induce protective immunity to block the penetration and replication of the pathogen into the mucosa or invasive, particularly in case of the invasive bacteria. So in this case, you require additional antibody, that is the serum IgG or the systemic IgG, not secretory IgA only help to prevent such kind of the infections. Okay, let's discuss about the whatever the vaccine available in the market and what lesson we learned with the help of this vaccine. So currently in market we have a two type of the vaccine available, one to treat the cholera and another one is to treat the typhoid fever. So for cholera, there are three internationally vaccines available in the market, and among three, two have a similar formulation and one has a little bit different formulations. Among these two different formulations, one is for the parenteral purposes and two is for the oral purposes. Typhoid fever vaccine also have a two type, one is for parenteral and one is for the oral. So oral cholera vaccine, which are available in the market internationally, and emphasize on the internationally, it's produced by the Dukerol company and another one is produced by Sancol and Ivuco. So this vaccines having the composition having the killed whole cell bacteria. This are prepared by using the monovalent Vibrio cholera O1 and bivalent is Vibrio cholera O1 classical strain, which are responsible for causing the pandemic long back, and another strain which are currently very found in the certain country, that is O139. Now, the Dukerol also has additional recombinant CTB component along with the whole killed vaccines. Among these three, the another one vaccine was also developed, but it was abandoned in 1917 because of these vaccines are not able to generate the gut mucosal response and it is an injectable base. So by observation of these three vaccines are that the protective immunity against the cholera are mainly mediated by locally producing antitoxic and antibacterial secretory IgA in the gut. This as IgA or secretory IgA binds to the B subunit of cholera toxin, that is the cell binding part of the cholera toxins, as well as O1 of the oligosaccharide antigen of lipopolysaccharides or cell membrane. So antitoxins and the antibacterial antibody both cooperate synergistically for providing the protections. The another example is the vaccines against the Salmonella typhi. There are three vaccines marketed internationally. Two are the injectable and one are the oral. The injectable formulation has a VI polysaccharide antigen conjugated to TT toxins and it is more effective compared to other two. And another one is a VI polysaccharide alone unconjugated vaccine. And third one is a live attenuated type 21 oral vaccines and it's given as a enteric coated capsules. So it is believed that the protective immunity against Salmonella typhi is not only mediated through the secretory IgA, but three types of the immune response. For example, secretory IgA required to prevent uptake of the pathogen across the intestinal barrier. The serum IgG required to prevent the systemic spread of the organism, and T-cell mediated immunity required because this Salmonella also are intracellular pathogens. So to kill the intracellular pathogen and to stop the further spread of this disease, the T-cell mediated immunity is also necessary. Now, Although the typhoid vaccines, injectable typhoid vaccines are effective, but still we believe that the oral mucosal vaccines are more prominent to provide the protections compared to injectable. There are several reasons why we require oral mucosal vaccines against enteric pathogen. One, the mucosal vaccines prime better immune response in compared to parental vaccines because as they mimic the natural infection or infections. They induce the specific secretory IgA and T-cell mediated immune response in the gut. And the memory lasts for several years in contrast to the clinical infection, particularly in case of the secretory IgA. Mucosal immunity, oral mucosal vaccines able to induce both humoral as well as cell-mediated immunity at mucosa and systematically. There is no interference from the persisting maternal antibody in the infant. So this kind of the vaccines are best suitable for the pediatric population. It is easy to administer, does not require any trained medical persons. There are less risk of transforming the infection because it's a needle-free, allows simplified production. So this oral bacterial vaccine, which are either formulated using a whole cell bacteria or kill bacteria, does not require extensive purification, while in case of the subunit vaccine we require extensive purification because it's given by the intraperitoneal route. It also speeds the vaccine development and immunization program. For example, in 1996 121 million children were immunized with the oral polio vaccines over a 2-day period. However, despite of such advantage, we have only handful oral mucosal vaccine available in the market. Whatever the oral mucosal vaccine available in the market are either prepared from the whole inactivated bacterial cells or live attenuated bacterial cells. There are certain novel effort which can make the oral vaccine by using a subunit component by certain chitosan and liposome mechanism or virus-like mechanism, but still it's not in a market. Live attenuated whole inactivated cells also provide a mucosal immunity, but they have a problem. For example, in case of the whole inactivated bacterial cells, it has a low efficacy because it not replicate once inside these cells. It requires multiple boosts. It need additional adjuvant, and it is also serotype specific because this vaccine are generally prepared by using a one bacterial strain. So it's provide only protection against a one bacterial strain, not other strains. Same thing with the live attenuated bacterial cells, although it does not require additional adjuvant and multiple booster dose in most of the cases, but there are always a chance of the reversion of the pathogenicity. So the stability as well as safety concern is always remain with the live bacterial cells. So when we talk about the enteric bacteria, most of the target group is the children below the 5 years of the age. So if we want to prepare a vaccine for the mass immunization and that to be for the pediatric population, the ideal vaccine should provide a mucosal immunity along with the systemic immunity. Needle-free approach is most suitable. It should be safe and efficient and cost-effective, and should provide a protections against a multiple strain. For example, Shigella has a more than 50 serotypes. So if you make any vaccine using one serotype, it might possible that it will not provide a cross protections. Okay, so now I would like to discuss some of our work which I have done in the B.V. Patel Pod Center. It's my previous institute. In B.V. Patel Pod Center, me and my team has designed specific oral mucosal vaccines against Shigella by using a food-grade bacteria, that is the Lactococcus lactis. So why we have choose a Lactococcus lactis? Because it's one of the safe alternative. It's a gram-positive, non-sporulating, non-invasive bacteria, and it's generally regarded as a safe bacteria. It also has an immunomodulatory effect, so you don't require to supply it with or formulate it with other adjuvant. It's enough to elicit the adjuvant effects. The potential of the Lactococcus lactis as a mucosal delivery vehicle for vaccinal purpose is widely reported. So our strategy is to make the recombinant Lactococcus lactis expressing a conserved protein. We have selected outer membrane protein A and give this recombinant Lactococcus lactis as a in a water or any formulations by orally. Once human or any host receive this vaccine formulations, this Lactococcus lactis enter into the gut and through the gut enter into the ant cells, the similar mechanism where the most of the enteric pathogen enter and create the immune response. So it will again doing the same kind of the mechanism and generate the same kind of the immune response. We have selected the outer membrane protein for expression of into the recombinant Lactococcus lactis. The one reason is outer membrane proteins are the conserved proteins among all the Shigella. We also found that this protein also have a conservancy with other Enterobacteriaceae. So we predict that this vaccine will provide a protection against multiple Shigella strains. We have identified two outer membrane protein, OMP-A and OMP-C, and we also done analysis like epitope and conservation analysis. And after that, we have evaluated for its immunogenicity by simply injected into the mice, and we have found that OMP are able to induce a good serum IgG level. And after confirmation with the immunogenicity, we have expressed OMP-A into the Lactococcus lactis expression system. After confirmation of recombinant Lactococcus lactis expressing the OMP-A, we have immunized mice sorry, with recombinant Lactococcus lactis expressing OMP-A, and by two routes, one is through oral and another one is through the nasal. Both are the mucosal route. And both are successfully generate the IgA as well as IgG level, but our findings suggest the oral route is better for generation of the secretory IgA and serum IgG level. So we have finalized the oral route for our further study. The main limitation for the vaccine development in case of the enteric pathogen is to mimic the exact host pathogen interaction using the animal, because whatever the pathogen causes the disease in human, they don't cause the disease in the animal. So it's very difficult to make a mouse model or any animal model which resemble the human-like syndrome. Similarly, in case of the Shigella also, previously, most of the people have used either rabbit ileum ligated loop or guinea pig or pulmonary lung model in the mice. But as pulmonary lung model in the mice just resemble the inflammation condition, not resemble the symptomatic condition as a human like shigellosis, we have tried to develop the intraperitoneal model, and we have successfully developed shigellosis, human-like shigellosis model in the biopsy mice, and which is evident with the feces, loose feces, wet anum, the shortening of the colon length, which is one of the hallmark of the inflammation, and bacteremia. Now, by developing this model, we again challenge with the, we challenge immunize mice, immunize with the saline wild type Lactococcus lactis and the mice receiving the recombinant Lactococcus lactis expressing OMP-A with the Shigella flaccinare. And what we have observed that all the mouse are successfully elevated the serum IgG level on the day 63, as well as anines or the significant increased level of the fecal IgA was also observed with increase or the significant increase in the interferon gamma and IL-10 level and also. And the immune, mainly we have found the Th1 ratio. Now, vaccinated animals are also protected from the shigellosis-like symptoms. The vaccinated animal does not develop any symptoms like unvaccinated or saline control or wild control animal develop. Now, we also correlate the symptoms or these tissue damage with our protections. So what we have done by examining the histopathology analysis, we have do the analysis of the percentage of the cell death in epithelium, lamina propria, crypt, and mucosal layer, and then make a table and correlate with the high level of IgG, secretory IgA, interferon gamma, and IL-10 level. And we have found that the higher level of the IgA have a low amount of the tissue damage or the pathogenicity. In case of the all four, IgA, secretory IgA, interferon gamma, and IL-10. And we have published this result in the vaccines. Our group also has work on the DNA vaccine, but because of the time limit, I am not going to present. So this work mainly done by the Brugu and Russia. And currently I have in my lab the Bhargav and Divya and my collaborator Vaino Patel, who are helping us in the various animal study. So thank you.
Thank you, Dr. Preeti, for a very lucid presentation, and especially your slides were very, depictive and very informative. Now we'll go to the questions posed by the participants. Aryendra Arya, his question is, why infections like leishmaniasis, mucocutaneous, and visceral form hypergammaglobulinemia, but they are low affinity and unable to neutralize parasites?
Frankly speaking, I don't have experience on the parasites part, so I don't want to comment on parasite part. I think Rajiv will will provide a better insights into the parasites.
So maybe the antibodies may not be very effective in neutralizing and clearing the infection probably, and that's why you do have hypergammaglobulinemia, but still it is not very effective. Possibly, I'm not an expert in the area, of course. So there is another
pathogen has a different mechanism. If we can classify it into parasites, bacteria, and virus, also they have a different. For example, in case of the enteric bacteria, also all four bacteria behave differently. So we cannot say the one mechanism which work for one bacteria can work for the another one.
Right. So here Shivani Yadav asks whether Lactococcus is not pathogenic, then why will it enter intestinal M cells to replicate and not cleared off by the immune system?
Right. So basically Lactococcus lactis is not a pathogen, but again, it's not a normal microflora also. It's something foreign to body, but it does not cause any immunogenicity into the body. Now, when it entered into the M cells, it degraded into the cells. But our purpose is to just hand over the antigen into the system. So I am not bothering about whether the Lactococcus lactis will remain live or not. Its function of the Lactococcus lactis is to just hand over the antigens to the antigen presenting cells at the dome of the follicle associated epithelium in the intestine.
It serves more like an adjuvant?
Yes, it's an adjuvant, and there are a number of studies done in which they have reported that Lactococcus lactis generally survive in the system with a 24 to 48 hours. After 48 hours, it will clear from the system.
Okay, so probably it helps in prolonging the antigenic persistence.
Right.
Right. So Sandhya Rani asks whether probiotics give protection on enteropathogenic infection.
Yes, probiotic always give a protections, but the thing is at what level? If the condition of inflammation and the pathogenicity is severe, then it will not help. You require some another kind of the treatment also.
Okay.
So based on severity.
So Michael Duraraj congratulates you for your wonderful presentation and for clarifying so lucidly and very educative presentation. And I agree that I was very impressed by your slides. So he has a question that how the vaccines are cleared from the system. Can you briefly comment?
Can you repeat the question? Sorry, Kalpana.
How vaccines are cleared from the system?
Okay. So basically, again, it's the same kind of the things. Lactococcus lactis will not survive. It just hand over the antigens to the antigen presenting cells, and then it will give the memory cells. Okay, it induces the memory cells. So when any pathogen come into the system, it will generate the memory B cells, long life cells, and protectors.
Right. I think you just spoke what I was about to ask you finally from my end, that do your the vaccine that you tried out and recently published, whether it does show good memory development.
Yes, it shows. But again, it's very preliminary result. There are number of things needs to be done. So,
any questions from any other panelists? Now, I see there's one more question from Sushil Sahu. Is it good to take all types of vaccines available in the market? Why is there cross-vaccine protection?
no, no, definitely no. But yes, for example, similar kind of thought also come in my mind. For example, in case of the Shigella or in case of the Salmonella, where you have endemic country which are already infected with such kind of things, and they have some kind of the immunity. So when you give vaccines to these people through oral, they don't establish a proper immune mechanism. So for that, maybe you require some another type of vaccine. So combination of two maybe help, like priming with the other thing. So means whole cells bacteria, then priming with the protein subunit vaccine or priming with DNA vaccine might help again.
Thank you very much, Dr. Preeti Desai, for clarifying the queries posed by the participants. And with this, we come to the end of the session. And I request Dr. Arora to take forward the next to the next talks. Thank you.
Thank you.
Dr. Arora, Dr. Bulan. So I just had a talk with Dr. Arora, and he suggests that we break for the next session and return back at 4:45. Thank you all. So the next session will start at 4:45.
Hi, we're very sorry we lost the connection. So the next session would start at 5 o'clock, please. There was a connection error, so we could not
Dr.
really
Gul Anand,
catch up.
Dr.
So,
Anand,
am I audible?
Dr. Anand, I had a word with Dr. Sunil Arora. He says that we will begin the session at 4:45.
Great, great, right.
Yeah.
So we'll.
And I have put it into the chat box also.
Yeah, thank you so much. There was a connection error, so lost the connection completely.
Sure,
We're
sure,
sorry
sure.
about that.
Yeah, thank you.
Thank you so much. Thank you all. हेलो नहीं कर दिया मेरा ना नेटवर्क हो गया था मैंने कर दिया अभी अनाउंस किया मैंने उसे सॉरी बोल दिया कि नेटवर्क हो गया था तो कनेक्शन वाज लॉस्ट और